Hello and welcome to the first day of MD&DI's Focus on Fundamentals course, How to Develop a Risk-Based Biological Safety Evaluation per new U.S. FDA guidance, sponsored by Nelson Laboratories. I'm your host, Tanya von Gremkow, Program Manager at UBM Americas. Before we get started, I have a few items to get through. First, the slides will advance for you. If you would like to download the slide deck, click on the Today's Slide Deck under Educational Resources. Please ask questions at any time during the presentation by typing your question in the question and answer box. Submit questions as they come to mind, and our speaker will address them during the Q&A session as time permits. The public chat box is for you to share thoughts and ideas on today's topic and with each other. And finally, at the end of the course, we will ask you to complete a short survey. Please take a moment to fill this out, as your feedback is important to us and will provide valuable information on the subjects covered in this course, as well as how we can improve future broadcasts. Now on to today's class. Develop a Biological Evaluation Plan, or BEP, presented by Thor Rollins, Senior Scientist at Nelson Laboratories. Thor is a certified microbiologist and specializes in the selection and conduct of in vitro and in vivo biocompatibility tests. He actively speaks on biocompatibility-related topics through Nelson Labs, external seminars, webinars, and trade shows. He is a participating member on all Association for Advancement of Medical Instrumentation, 10993 ISO committees, and plays an active role along with the FDA and regulatory committees, developing standards, discussing biocompatibility methods, and voting on changes to those standards. As one of a select group of experts in the industry, Thor's participation in the committees offers him insight on industry changes and helps prepare clients for changes in testing. For more on Thor, please see his bio under Featured Speakers. Before I hand it off to Thor, we have a poll question, which will appear to the right of the slide window. Let us know, how does your company address biocompatibility? Select one of the following answers. Do you checkbox the testing in the biocompatibility chart, talk to a CRO for advice, ask the FDA what they would like to see, or identify the risks to the patient and test to fill in gaps? So we'll give you a few seconds to answer this question, and then I'll hand it over to Thor, and he'll publish some results later in the presentation. So let me give you a few more seconds here. Get a few more people to answer. So just check one of those. If you can't find the window, it's off to the right, next to the slide window. Okay, I am going to close the poll now and have Thor start his presentation.
Great, thank you so much, and thank you for everyone for taking the time to sit in and discuss biocompatibility with us. It's something that I am very passionate about, so I tend to speak quite a bit about, but I know that for some of you, it's not as a loving affair, it's more of a hate affair with biocompatibility, so thank you for the time. The reason we wanted to do this webinar is we're seeing a very large trend or movement in the industry regarding biocompatibility. And we wanted to kind of get information out there about the new approach on how to evaluate biocomp. This three-part seminar or webinar will be looking at these three separate phases of a biocompatibility plan or program. The first phase that we'll be talking about today is how to develop a BEP or a Biological Evaluation Plan. What goes into that plan, what kind of steps goes into developing a plan, and how to best make sure that that plan is correct for your device. After you develop the plan, tomorrow, Dr. Campbell will go into certain tests. Now, this category can be much longer than an hour webinar can be, so Dr. Campbell will be specifically focusing on the leachables and tox evaluation of a test evaluation. And then on Thursday, Audrey Turley will be looking at the biological evaluation report, basically the summation of your plan and your outcome of your test and how to develop that and what goes into it. But today, we will be focusing on that BEP, that Biological Evaluation Plan. So this plan has certain steps that we want to evaluate, and it really goes into what is a risk-based approach. What kind of items that we want to evaluate when we're looking at our devices, the materials and the processing and the risk associated to the patient. But a lot of times we think, why now? What's different between this approach than in the past? What has been the catalyst for this change going forward? And there's been two main things that's happened recently that's really stimulated this type of approach with biocompatibility. The main one happened in June, and that's this guidance document that hopefully you all are familiar with. This guidance document was released by the FDA in June. It was a finalization of a draft document that came out in 2013. And when this is my my evaluation of that document in the photo. It's quite a good document. I actually really like it. I think the FDA did a really good job of spelling out what they believe is their approach to biocompatibility. They did a webinar about a month ago covering this new document that you can access if you are curious about it. But in this document, the overall theme of this document is about risk. And when you start looking at a medical device used in the clinical application, really that's what it comes down to, is what is the risk of that device. I feel like all of us have gotten into this industry to improve patients' health and to make life better for individuals that are suffering. And I think that's the main reason that all of us are working in this industry. Well, the big risk to that is these medical devices are made of foreign material, and we're putting it into the body, so there's always risk. If someone tells you that this material has been used historically forever and that there's no risk associated with it, that's not true. Anytime you introduce a foreign material to the body, there's risks involved. What we have to do is identify that risk and then evaluate to see what gaps are there, and then do a risk-benefit ratio. Is a benefit of the device outweigh whatever risks are associated with the device? And that's what this whole biological program entails. When you develop a BEP, you fill in gaps with testing, and then you sum up with a biological evaluation report. You're really taking that whole concept, that holistic view of biocompatibility in account. And that's what the new age of biocompatibility is going to be. For those of you that may not have had the pleasure of going through the 60-page guidance document from the FDA, here's the link. So if you are not busy tonight, I recommend that you sit down and go through this document to really familiarize yourself with what the current applications of biocompatibility are from the FDA. In this document, the FDA guidance says the guidance document includes the use of a risk-based approach to determine if biocompatibility testing is needed. And that's a quote from this guidance document. It says if biocompatibility testing is needed. And that's the main thing that I hope that you get out of this presentation, that biocompatibility is not a checkbox mentality. It should not be a checkbox mentality anymore. We talk to dozens of customers every week, and to help them evaluate these plans or come up with these plans. And it is a big change to change them from checkboxing how they've always done their biocompatibility to go into a risk-based approach to really evaluate where their needs are associated to their device. So to help encourage that, that's what this presentation is going to cover, and how to evaluate that risk. If you are not familiar with ISO 14971, you should also familiarize yourself with that document. This is basically what the new ISO 10993-1 is applying. So just so you know, 10993-1 is in draft right now, going through with comments. And in that draft, it is very heavily associated with risk-based approach and very closely associated with ISO 14971. And in that standard, they define risk as the combination of the probability of occurrence of harm and the severity of that harm. That is the essence of what this BEP is. It's to evaluate the probability of harm and then the severity of that harm that could occur. And when we do that, it's a little bit different from some of the other evaluations that are in 14971. but the spirit of it's the same. Our probability of occurrence goes into materials and processing and how it's used, and then that goes into an evaluation of the severity and impact of that device. That's why I love this picture. I got this from a friend of mine, Dr. Ed Rivardi, and I love it because it symbolizes the risk versus reward approach of a medical device, where that mouse wants that cheese, and he realizes the risk of the trap, and he's trying to evaluate to see if his helmet will be a gap in that risk to be able to successfully get the cheese. And that is what the BEP basically entails. So what is that past approach? What is it that we've done historically? I think most of you are familiar with this table. This table resides currently in the guidance document from the FDA. It was also part of the G95 document that was released back in 1995, and it's also in an appendix in the ISO 1093-1. And historically, it has been used as the checkbox for evaluation of tests that you need to perform. How we currently use it is we still use it, but how it's currently used, it's no longer used as that checkbox. So you don't go through and check, 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 check. What it is the endpoints that you need to evaluate based off of the use of your device. So you can still go through and use it, and we'll go through this a little bit more in my presentation on how to evaluate your risk of your device. But this is still a useful tool to evaluate what endpoints need to be addressed. The difference is those endpoints are not necessarily testing. You may be able to evaluate those endpoints with different parameters or different ways to evaluate. So that's the difference with the current revision of the guidance document from the FDA and the current draft of 1093-1. This chart has been lessened in its importance. It's no longer that checkbox, that say-all of biocompatibility that it has been. So with this past approach, the problem with that past approach is it really is that black box, and it takes out all the necessity from you as manufacturers or material suppliers or whatever your position is. It takes away your responsibility to understand what you're putting into a patient's body. In the past, you used to just get a material, run it through these in vitro and in vivo tests, and depending on the outcome, that would be your safety assessments. You would say, we passed all of biocompatibility, therefore this device is biocompatible. And it really did not allow you to understand or push you to understand the impact of these materials or the processing involved in this manufacturing of these materials. So it was kind of a lazy approach. The more we've got to know these animal testing in the decades that we've used them, the more we kind of see that they may not be the best predictors of patient health, at least not that what we thought they may have been decades ago. So just putting them in the suite of testing may not solve that risk problem that we are all concerned about. So this is the past approach. You would just evaluate that contact, you would do the testing, and you submit it to the regulatory authority, and you would almost no one had to have any background in safety or biocompatibility or toxicology to do this approach. The new approach, and this is kind of speaks to it, this slide, where you didn't have to understand that material's impact on the body, you didn't have to understand the testing at all. You just, as long as you got good results, you were fine. So I always use this as my example. Some of you may have heard this before, but it's my car example, and I use it to share kind of a real-life example to me on the different types of approaches. So on the left hand, we have a 1967 Mustang. That was my very first car. I loved that car. And it got me from point A to point B. Sometimes it got me from point A to point B very, very fast. The problem with that car is it's not very efficient. In fact, I could actually watch the fuel gauge start to go down when I rode in it. And so even though, and it didn't have power steering, so it had a steering wheel the size of a bus, and the brakes were drum all around and didn't stop very good. And so the car just, even though it got me from point A to point B, and I did love that car, it was not efficient. You did not want to go long distances in that car. It was not, you end up smelling like exhaust and speed up. The car on the right is what I currently drive. The version of the car I currently drive that I want to drive. It's a Ford Focus. Now, that car is, even though it's still a car, has 4 wheels, has an engine, still gets you from point A to point B. The efficiencies are much greater. It's much more comfortable to drive. It's more safe. There's a lot more benefits that goes into it. And this kind of sums up the biocompatibility approach. If you want a checkbox right now, especially with the FDA, Europe right now will sometimes ask you to justify doing unnecessary animal tests. But if you want to checkbox your testing, that's still a way to get from point A to point B with a lot of regulatory agencies right now. That is changing, but right now it's still a way to get from point A to point B. But it's not very efficient. It really can cost you a lot more time and money than this risk-based approach. I am going to publish the results of the poll now so we kind of get an idea of how people are evaluating biocompatibility. As you can see, when we look at those 4 questions, and this is very common, and I'm actually kind of glad to see that the majority of the people identify risk to the patient and test to fill gaps. But the second most answered question is the checkbox mentality, which once again is something that we see when we evaluate biocompatibility. I am glad that people talk with CROs for advice. That is something that if you struggle with a very complicated device, it's something that's good. I wish that we would see more people ask the FDA what they'd like to see, and we're going to get into that a little bit more. My friends at the FDA have been asking me to make this a priority or emphasis when I talk to my customers, that they would like to get involved, especially when there's a risk-based approach, to be able to make sure that all those gaps are filled to their satisfaction. So this is very interesting. It's something that, like I said, is very similar to what we see in the industry. And hopefully at the end of this presentation, that 34% of you, the 24 of you that answered checkbox, we get you to move a little bit more down into that risk-based approach. Okay, so this chart, this figure, is figure 1 out of ISO 10993-1. And that's the current chart in there. It's a great chart, and the reason why it's a great chart is because this chart allows us to evaluate the risk. These are really the steps that you need to take in your BEP. The first one, and it's something that I really see sometimes people struggle with, is does your device have intended patient contact? If it's a no, ISO 10993 does not apply. What that means is that if there's portions of your device that do not have intended patient contact, then you do not need to evaluate through 10993 the biocompatibility. You can still evaluate risk if you still want to evaluate risk of that material or device, but as far as ISO 10993 goes, it does not apply. The new guidance document that was released by the FDA in June does have a little bit of a different twist on this. If you read in the beginning, it talks about patient or direct or indirect contact. And it does put in a little bit of a stipulation as far as clinician or user contact. And what it says in there is that if you are intended to contact the user or clinician of your device, and that clinician or user is not protected or gloved or has some kind of protective gear on, then biocompatibility should also be assessed for that user. So this goes to maybe exam gloves or some other devices that may not have intended patient contact that would be in contact with the clinician skin, and in that case you would need to evaluate it. The one thing that I can think of right off the bat with this that would be affected is some kind of mask guard for a surgeon. That headband that goes around the skin, that headband doesn't have intended patient contact, but it does have intended clinician or user contact, and so that headband now would need to be evaluated for a risk-based biocompatibility approach, where before you may not have been. So let's look at this chart. So let's assume that all of your devices and materials that you're looking to evaluate has at least intended patient contact. I do also want to point out here that there is certain cases as far as failures go on devices or risk or worst case examples where materials that may not have intended patient contact will have patient contact based off of that failure. And if that failure is a realistic failure, not a ridiculous failure, but a realistic failure, then you should also have a risk-based approach for that material also. But once we have that decision that it does have that kind of contact, then you go through these questions. And these questions are designed to evaluate the risk of that material or that process. It talks about, have you used these materials before in other devices? Have they been processed the same? Are there have you had experience basically with this material? And if you've used these materials in similar applications with the same processing, then you can go into a biological evaluation and be done. So obviously you have to be able to prove that or state that to a satisfactory level to a regulatory agency, but we've been very successful in this type of approach when we've been able to use a device that manufacturer produces and has the same processes and the same intended use. It's a fairly easy justification. So a lot of times that's not the case, right? There's definitely devices that are manufactured that you do not have that experience, that much detail on, but you're not necessarily destined to do testing at that point. You then go down to the next line, which talks about chemistry. It really goes into filling some gaps with chemical evaluation. You can maybe do some leachable extractable data. You can do some chemical analysis to really try to understand the leachables that are coming off your device to make some kind of assumptions. Only when you cannot do a material evaluation or a process evaluation and you cannot fill in those gaps with chemistry do you go down to the third line where the second box over where it says perform the testing per the annex, and that is that chart that everybody uses. So if you see that, the chart is actually one of the last things that you need to do to evaluate biocompatibility once you've exhausted all these other means of evaluation. And that's really the approach that we are seeing regulatory agencies start to come in line with. I mean, like I said, if you read that guidance document, it really spells out this risk-based approach. It really wants you to evaluate, and it gives you certain ideas on how to justify out of testing based off of materials that have been used before. It even brings in master files and what information should be in master files. It's a really good document. So when we're looking at biological evaluation plans, and remember, these are really just plans that evaluate the risk of these devices to the patient, to be able to spell out where your gaps are and how to and identify how to evaluate those gaps. So what should be included in these plans? Well, the first thing is we still need to talk about the device. We still need to evaluate its intended use, how long it's in contact with the patient, where it's in contact with the patient, what population is it exposed to, is there a special population, neonates or infants, that we may have more risk for. You know, all these questions need to go into as far as the intended use of the device. The next thing we have to look for is possible misuse. You know, will this device be used with your knowledge in a way that you did not expect it to or intend it to, or maybe just didn't put in your IFU because you didn't want to evaluate it? We have to look at that possible misuse of that device and the impact of that. Remember, we're looking at all the risks. And we talked about failure scenarios. Devices fell. Pieces fall into surgical sites. Balloons burst. These things happen. We deal with them every day. And so it doesn't mean that you have to do a full-blown biocompatibility checkbox assessment on something that may or may not contact the patient. What it means is in this plan, you need to address possible failures that could happen, that you feel is a realistic possibility. And then you need to evaluate that failure by looking at the materials and the processing and the risks of the patient with its use, and be able to come up with that risk and how to evaluate that risk. So this is the next step that we will include in our plans, material makeups. And sometimes this is much easier than other times. We've been through this quite a few times, and sometimes we can get great information from suppliers. Sometimes we get just an MSDS, which, trust me, does not give us enough information to make an accurate and adequate material review. I do also want to say here there are things out there like medical grade, USP class 6, non-cytotoxic, all these things you can get on these materials that you can include in the plans. But don't be fooled. Become educated on what this really means. Material grade, as of right now, there is no criteria around what is called medical grade. I've seen medical grade material that have had all the biocompatibility done on the material, and I've seen medical grade material where only it has a dedicated line that only that material be made, but no testing was done. So instead of saying, oh, it's medical grade, we're good to go, really delve in to see what it constitutes those claims. A USP Class 6 is a great claim, but it does not include cytotoxicity. And it's very limited to, it's supposed to be for raw material plastics for container closures, but we see lots of medical device manufacturers or raw material manufacturers try to use it, but be careful, it does not include cytotoxicity. So all these things have to be evaluated on your materials, previous use, what changes may happen during the process, what impact does sterilization have on it. That's the next checkpoint. Believe it or not, sterilization can have a huge impact on some materials. What we've seen is EO, ethylene oxide, has a more limited impact. It has heat and humidity that could have an impact in materials and its leachability, but the gas itself doesn't necessarily have as big of an impact as radiation does. Radiation, especially to polymers, can have a huge impact. I have seen devices that have failed the cytotoxicity test, and then once they get irradiated, they pass. And I've seen other polymers that have passed before radiation that have failed afterwards. So there's not a strict rule that we can tell you that if you irradiate it, it's going to make it better or worse. It's just going to change it. So that means that we have to evaluate the risk of this device post-processing. Cleaning, these detergents that could be toxic, oils from the manufacturing line, colorants, which we'll get to in a minute, all these things could have possible impact to safety and all need to be put into your plan as a possible evaluation endpoint. We talked about special populations. Are there certain people that are contacting this device that have special considerations? So how do we evaluate the intended use of the device? This is where we use that chart. Like I said, we still use that chart that everyone uses. The difference is we do not checkbox that chart, right? We use those endpoints and we say, okay, my device needs to have an assessment done for sensitization. Have I used this device before? Have we seen sensitization happen? Do I have something in my process that's a known sensitizer? These are things that we want to evaluate to look at risk. This is also where we evaluate that misuse and failures. So I wanted to bring this up. This right here is the chart that everyone's familiar with. How we use it is on the left. We have body contact, right? So we have surface devices, external communicating devices, or devices that are inside the body and outside the body at the same time. And then we have implant devices. So all these endpoints need to be evaluated. The subcategories are pretty self-explanatory. The only one that some people get confused a little bit is between blood path indirect and circulating blood and tissue bone dentin. A lot of people feel like when they're in tissue, there's a lot of blood present, and they feel like that might be blood path indirect contact. Remember that these ISO committee members, they are aware that when you're in tissue and bone that there's blood present. What the blood path indirect and circulating blood, what the difference between that one is the tissue is that hemocompatibility. And that hemocompatibility really focuses on impact from circulatory contact. So you look at hemolysis and thrombogenicity, things that are not that concerning when you're in tissue contact, more concerning when you're either direct or indirect. So the best example for an external communicating device with indirect blood path exposure is an IV bag, where the IV bag sits outside the body. It has saline solution in that bag or whatever solution it is. It could be drawing leachables or chemicals from that bag, plasticizers, whatever it is, and those are being indirectly delivered to the circulatory system or the blood path. So that's a blood path indirect. The IV bag's outside the body, communicating to the body, and circulating blood is something directly sitting in the material sitting in that blood flow. So that's the difference. And then you look for contact duration. You look at anything less than 24 hours contact is A, 24 hours to 30 days is B. Anything over contact with the body for 30 days is considered permanent implant or permanent contact. This is also important to consider repeated exposure. So a device that's in contact with the body for only 15 minutes may be limited unless that patient uses that device every day for the rest of their life. Then we have to add up those 15 minutes every day to get an idea of the permanent exposure of that device. And actually, when you look at risk, A lot of times those devices carry a more of a risk than some metal device that sits in your body for length of the lifetime. Because when you look at most devices' extractable profiles, the majority of those compounds get extracted out within the first 72 hours, and then it starts to taper off down into a small amount. So if your biggest impact is in that first 72 hours, 24 hours, whatever the device may be, so if you're getting a new device every 24 hours, you're getting a brand new peak or dose of the highest peak over and over again compared to something that may be lessening the amount of chemicals being exposed. So those are things you have to evaluate when you're looking at the risk of your device. I wanted to point this out too. This is the chart in the new guidance document from the FDA. I have some arrows there that are some differences from this chart compared to the latest ISO 10993 chart. The main differences are they've added material-mediated pyrogen. So before, that was kind of an understood test if you had to do acute systemic toxicity. Most labs would tell you if you had acute systemic toxicity category marked, that would both be an acute systemic tox test and a material-mediated pyrogen test. I think the FDA just got tired of telling people that and made two separate categories. So now we have two separate categories, plus that allows us to be more focused on what is needed for pyrogenicity as separate from acute systemic toxicity. So I think it was a good move. They also put a little bit more emphasis on reproductive toxicology and degradation in that chart. The other thing that's important, and this is probably the most important thing to realize, is the FDA chart in the G95 document always had circles. And those circles were additional evaluations that may be considered necessary based off of your FDA reviewer. So basically, if your FDA reviewer thought that that material may be scary or something like that, then they would ask us to do those tests. The new chart, if you look at it right here with the O, it says additional FDA recommended endpoints for consideration. And if you look at the X's, it says ISO 10993 endpoints for consideration. Now notice they didn't say testing, right? It said consideration, but the FDA has considered those circles the same as the X's. Really the only difference is the circles are just for the FDA and the X's are for ISO 10993. So that is a huge potential impact in the industry because a lot of these tests have never been looked for before. For example, on this slide, if you look in the implant with a blood or tissue permanent, at the third to the right there is carcinogenicity. Carcinogenicity testing can be very, very expensive and long. We're talking millions of dollars. It takes years to do. So not many medical device manufacturers perform that test. And in the past, it's kind of been one of those things that people just forget about. Well now, and we've actually seen this from the FDA is asking you to evaluate carcinogenicity. And you can't just use genotoxicity as an endpoint to evaluate carcinogenicity. You need to do a carcinogenicity evaluation. That does not mean testing, or else labs would have a lot more revenue. What that means is that we need to find a way to evaluate the carcinogenicity potential of the device. It could be material evaluation. Most likely it has to do with what Sarah will be talking about tomorrow, which is extractable, leachable, and a tox assessment. We've seen FDA come back with permanent implant devices saying, where's your carcinogenicity endpoints? Please provide what they call a TE or TI value for carcinogenicity for these endpoints. That's where that chemistry and tox evaluation for tomorrow will get you. What you need to do for your plan, though, is have those endpoints included in your plan, all those circles and X's, and evaluate in your plan how you're going to address those endpoints, either through chemistry, material evaluation, whatever it is. That's the important part about the biological evaluation plan. I do want to mention one other thing here. The new ISO draft does have some additional things in this chart. It has a lot more emphasis on chemistry. In fact, it's actually going to include chemistry in the chart. So for certain devices, you are going to be required to do a chemical or material evaluation. It also is going to have a little bit more emphasis on packaging of your materials. We're not asking you to test packaging materials separately, but you need, once again, to evaluate the risk of that packaging to your device. Does it transfer? Is there melt? We've seen plastics melt onto a device or transfer inks from a label. These kind of things need to be evaluated. And it may be that your testing may have to be packaged in its final configuration through all its processing to evaluate any potential transfer. But those things should be spelled out in your biological evaluation plan. Okay, so the main thing that goes into your BEP that a lot of companies are not doing right now is this material evaluation. Really evaluating what is in your device. And we've had companies that have sent in biocompatibility that went down to Home Depot to buy some stainless steel. We would not recommend that. Nothing against Home Depot. They're a great company, but not so great as providing medical device material. So this helps us evaluate what really goes into a medical device and the risk associated to it. These are the challenges that goes into a material evaluation, and believe me, there are challenges. The first thing is, and this should be a no-brainer, but like my example just a minute ago, it's sometimes not. We have to understand our product. We see engineers understand the materials to a great degree as far as how they function, how hard the material is, how it needs to be stiff, and these kind of endpoints they understand completely. But when they get to the risk from a safety standpoint, there is no evaluation. There's no understanding. That's what we need to do, is to understand that product. We also have to understand the changes that could go through in its life cycle. And by its life cycle, I mean its process. So the risk associated with that device could be different depending on its processing or where it is in its process. You're looking at a same device that may have to add an additional cleaning step to it after it's been approved. Does that change biocompatibility? We had one recently, it's been a couple years ago, that changed locations. And when they changed locations, same device, same employees, same even machines, but when they changed locations, they started failing cytotoxicity. When they performed a chemical analysis, they found out that it was a chemical that's been used in surfboard manufacturing, and their neighbor just happened to be a surfboard manufacturer. So there are things that could evaluate through the life cycle that could have a change or an impact to safety. You need to understand the leachable substances within your product. That is probably the biggest help, and I, like all of you, are looking forward to tomorrow to understand more about how to do that from Sarah. And the big thing that this can help you do is, can you predict the safety from one or only a few tests conducted on that product? Do you have enough information? Do you have enough from the chemical leachable perspective or from the material perspective to only do a few tests to evaluate that safety instead of putting unnecessary animal tests at risk? And then remember that in some products, that safety could change over the lifespan of that product. Some degradation devices or materials will degrade over time, and that means that product safety may be different on the end of its life cycle or its shelf life than at the beginning. So those things have to go into the material evaluation. The other thing that I want to kind of talk about here that does not go into like a leachable extractable profile is geometry. And this is important for you to know. Most of the testing endpoints that we evaluate, from cytotoxicity to sensitization to subacute toxicity, are chemical-based. So an extraction is done to look at what comes off the devices. There are some other endpoints, particularly hemocompatibility and implantation, that may have a chemical component to it, but more importantly has a geometrically important or component to it. So how that device is shaped, the surface characteristics of that material have a bigger impact in hemocompatibility and implantation than the extractable and leachable substances. So if you're changing the geometry of the device, depending on how you're changing it, you may not need to reevaluate genotoxicity, for example, as long as the processings all stay the same. But you may need to reevaluate your implantation and your hemocompatibility. So those things need to go into your product also, or your BEP. So all these things kind of go into understanding what your product is. The one thing that I want to talk about here as far as influences within 10993, these are all things we talked about, but I wanted to mention colorants. The FDA, once again, did a webinar, it's probably been a couple months ago, that you can find about colorants. And it's been a hot spot with the FDA. We have a little bit more, I should say a lot more knowledge on what we need to do with colorants now based off of that webinar. I would highly recommend it. But if you have a colorant in your device that contacts the patient, especially if that device is more than just a short-term contact, we have to have a specific section on that colorant. We have to do some kind of chemical leachability analysis on that colorant. We have to have a tox assessment done on the components of those colorants. You have to get that information from the colorant supplier, which is sometimes easier said than done. A lot of times, actually, we at Nelson Labs have to be the generator of that information. We contact the color supplier and get an NDA in place and actually get that information independently of you, because I guess they figure we can't reproduce that colorant like you guys can. So those, if you have a colorant in your device, pay particular attention, especially if that colorant is prolonged or long-term contact, or if you see particulates or color changes in your extract media with your biocompatibility. Those are your things your FDA are looking for. Okay, so when you're looking at material chemical characterization, which Sarah's going to talk about tomorrow, that is the first step. When you're trying to fill in what your BEP is, and you can't fill in everything with your material assessment, your next step would be to look at your chemical, and that could be physical, electrical, any of those properties, and that's per 1093-1. So after you get done with your material assessment, now you have to basically evaluate that from a chemical point of view. The most important thing about a material and chemical evaluation is it does involve communication with your materials manufacturer, but we cannot just evaluate those materials. And this is where a lot of people get in a problem. So they get a material from a supplier that may have a master file with the FDA with tons of implant history and data, and they submit that to the FDA and they go, we don't have to do biocompatibility, we don't even have to do a safety assessment, because our safety assessment is this material is safe based off this master file. The FDA will not accept that. And that's because biocompatibility is just not about materials, it's about what you do to them, including sterilization. So please keep that in mind when you're going through a BEP. You evaluate your materials, you evaluate your processing to evaluate risk. Those are your two main components of a BEP when you're looking at risk. The problem with this is it involves communication with your suppliers. And these are some of the things that I look for when we're doing a BEP from your suppliers. I do want to mention here, we've seen to get better success from a supplier when you're in negotiations for a contract than when you have business already done with them. So keep that in mind when you're setting up a new supplier. Try to get these things up front. If we have suppliers on the webinar, I would like you to be aware that you are going to be asked this a lot more. So you may want to come up with a plan to be able to supply either a dossier or something, these types of information to a supplier, and I can guarantee you that it may be to your benefit from a business standpoint to be able to market this type of information going forward, because a device manufacturer is going to need this information to be able to do these types of BEPs. We have manufacturing agreements. Most of the time when you have a manufacturing agreement is a, do not change or don't change anything unless you tell us kind of agreement. Be careful with those. We had an example a couple years ago where we were doing routine cytotox testing on a glass material that they purchased and were using in their device. So they did a raw material cytotoxicity on every lot. And for years they got zeros. And all of a sudden they started getting 2s. Now, 2s in cytotoxicity is still a passing result, but man, you go from 0s to 2, what's going on? So they called up the manufacturer and they said, well, per our agreement, we would always notify you if there's a change, and there's been no change. So they went down and audited, and they went to the cell that was making the glass, and the supervisor of the cell came up and said, well, I want to show you how good we are, how clean we are. I, the supervisor, have started mandating that everybody wear gloves during manufacturing. This person asked, what kind of gloves are you using? And they said, well, these are over here. And it was powdered latex gloves. And for those of you who are not as familiar with the cytotox test, latex is our positive control. So this is not something that they would have told you had changed, because it was not something that they felt like was a change in the process or in the material, but it could have had a big impact on biocompatibility. So please pay attention and really get detailed as far as your manufacturing agreements. Composition disclosures. We have to get to a point where people are not so scared about divulging information. I know that's easy for me to say being a lab, But in order to do a great material assessment, we have to know what's in that material. Because even though I hear it all the time, oh, it's silicone, it's polypropylene, it's PEEK, and they say everybody uses silicone, polypropylene, and PEEK. Yes, but not all silicones are the same. Not all polypropylenes are the same. They have different stabilizers, different additives, and so the toxicities can vary quite a bit between different types of silicones and materials. So just because it says that it's a silicone, it doesn't mean that you're done. You really have to know what is in it, and if that requires chemistry to do so, so be it. We also have to get whatever they're doing in process. This goes back to the latex, everything that's in their process and chemical disclosure. And like I talked about at the beginning, this does sound like a lot more work, but this is a one-time thing. Once you get this and you do this evaluation, think about the time and money that spends on the backside being able to justify out of some of this testing that we are very long and very expensive. I did knock MSDSs before. They are useful. It's just sometimes that's all we get, and it's not useful by itself. One of the best things to get is a device master file. This is something that they submit to the FDA to basically get a pass themselves from the FDA. Now, like I mentioned before, the new guidance document has a whole section on what a master file should entail. So don't think that a master file is just a pass to go. It is very useful, but they should include, any master file should include what that new guidance document has in it as far as what is recommended by the FDA. And then also remember that that master file is just for that material. It's not for what you're doing to it. So you still have to address the processing, but the master file is very helpful. Okay, so to kind of sum up what we've talked about today is this biocompatibility assessment is critical. It is needed. It's going to be, in the future, what everyone has to do. There will be a time, in my personal opinion, that regulatory agencies will kick back a checkbox testing approach. Because if you think about it, in their point of view, a checkbox really doesn't give them a lot of information. You pass these tests. Great. That doesn't really tell me a lot except that you pass these tests that are 20, 30 years old, where a material risk analysis with a chemical characterization gives a lot of information about what you're putting in your device, what processing it's going through, what are the potential risks, and how you identify those risks. So I really believe that once the regulatory agencies start to see these type of approaches, they will like these type of approaches. Maybe the most important thing about this, and this is the biggest hurdle for some companies, is it no longer can be done just by some engineer or regulatory person just looking at that chart. You know, that's the downside of this approach, is before you just needed someone to basically read a chart and do the testing. Now it does put a little bit more emphasis on having experts in certain fields of chemistry, material chemistry, a toxicologist, a biocompatibility expert. These types of people should be involved in this risk assessment. Sometimes companies don't have those people. That's okay. All the CROs out there do have those people. So you just can use your CRO to help you if you do not have the needed people, but those people need to be involved in this type of assessment. That also leads to that chemical assessment, which is probably one of the most important parts of an evaluation, especially if it's a permanent contacting device, because without that chemistry evaluation, you're going to be stuck trying to evaluate genotoxicity, carcinogenicity, chronic toxicity, reproductive toxicology through other means. So the chemistry becomes a much more important role in this new type. I do want to emphasize that this is a written plan. You can't just go to the FDA and say, we evaluated risks, these are the tests we felt like we had to do, and we did them and passed, and we're good. You really have to sit down, think through that risk, those materials, all the things we talked about, and put it on a paper plan, a document that spells out what is the risks, how you're going to address those risks, and have a signed conclusion that, you know, this is how we're going to approach our biocompatibility with this device, and have those people involved, those chemists, those biocompatibility experts, the toxicologists. I also want to make a point here that this plan is great if we submit it to the FDA, especially if you are trying to justify out of testing. because what you can do a Q-sub or a pre-sub with the FDA, which is an official submission. I've also had companies that have really good relationships with reviewers that they've done informal kind of pass-throughs. But this is great because you get their feedback on the risk. And yes, we really want to make safe devices. That's the number one thing we do this for. But the second thing, which is slightly less important, is to get it through the regulatory agencies. So without getting their feedback on these plans, really you're only addressing that first main reason, but you're missing out on the second one. And there used to be a time where the feeling with FDA reviewers where there's a big scary monster in the building, and you didn't want to talk to them. You just wanted to cross your fingers, close your eyes, submit your testing, and curl up in the fetal position and wait to see if you get anything back. Now, these pre-subs or Q-sub meetings have been so useful for a lot of my customers. They get good feedback. The FDA has actually had times where they told us that the testing is not needed, and which I know shocks some people who are not familiar with the FDA recently. But it is something that we recommend because you get good feedback from them. You have higher confidence that if your testing comes out good, then you have met those risks that the FDA have in their minds. So this is the BEP, the aspects of the BEP that we put in them, how we develop them, and hopefully this has helped you evaluate how to do risk on your device and come up with an effective plan. Now, are there any questions?
Thanks, Thor. That was great. If you have questions, please submit it now using the question and answer box. If we are not able to answer all the questions, we will share the remaining questions with Thor, and he'll respond directly via email. So let's get to our questions. The first question we have here for you, Thor, is our cytotoxicity sensitization, sorry, it's a lot of words, irritation tests done back at 2005 on the same dental implant finished good, and in parentheses it says same material and process, can be still applicable for a biological evaluation today? Is there an expiration date for data used by the company for the biological evaluation? That's question 8, Thor, if you need to reread that or have me reread it.
I got it. Great.
Okay.
And I'm sorry, us biocompatibility people love to make very complicated long words, so don't feel bad about it. So this is a great question. And as far as the expiry date, we do update standards, and in fact, the update relatively regularly, depending on how much argument happens in the ISO committee. But what we need to do, testing done in 2005, if it's done on the same material, the same processing, may still be applicable. What needs to be done is there needs to be a gap assessment performed on that testing to see if it still meets the current ISO guidelines. For example, irritation was updated in 2010 to where it went from a 2-rabbit to a 3-rabbit test, and it also changed how the scoring criteria was done. So an evaluation can be done to see if we can still use those results. Even if you used a 2-rabbit test, we still may be able to evaluate the results and make an assessment that a 3rd rabbit would not change the impact. And remember, these tests are meant to screen for possible clinical impact. So if you did these tests without any clinical experience, then yes, those absolutely has to be done to reevaluate that gap analysis. But if this device has been on the market for that long, there's a lot of good data that we can get from that to show safety besides just putting it into a guinea pig again. So all this goes into that risk assessment, BEP, those kind of information. So very good question. I hope that answered it. If not, you can email me and let me know.
Great. Thanks, Thor. So the next question we have is there an option not to do even one test if it is justified, supported by literature and experience? If yes, even if we talk about a dental implant made by titanium alloy and well-known manufacturing process?
Another great question, and the answer is yes, although it is a little bit more difficult to do no testing. But we have done it. You really have to get very detailed information for the material, your history with that material, like it's from the same supplier, nothing's changed, the processing you're going through is exactly the same, you look how it's used, all those endpoints, and if you can really talk to that, then yes, we can just do a paper approach, and that's been successful for those kind of circumstances. That being said, it also depends on what regulatory agency you submit to be completely honest. There are certain regulatory agencies that seem to be more lenient when it comes to evaluating animal testing in particular than others. So it does kind of depend on your regulatory arena, but if you have a good enough information and risk assessment, all regulatory agencies will take no testing. That being said, we usually always recommend a little bit of in vitro test here and there just to do some kind of evaluation. It adds to the assessment, and it's not that expensive or time-consuming. But in theory, yes, you can do no testing if you have enough information.
Okay, great. So our next question is, what if our component comes in contact with a drug that will then go into the body? Our product does not touch the patient directly.
Another good question. So this would be a classic example of an indirect contacting device. So let's say that's a vascular drug, then you would be an external communicating device blood path indirect, because what chemicals could come from your device into that drug, leach into that drug, and then be delivered to the body. So yes, even though you don't have that direct patient contact, you would have indirect patient contact through that drug. So a risk assessment should be performed for that type of exposure.
Okay, so we've got lots of questions here. The next one is, how would you suggest evaluating the risk for a device that has different types of exposure? In parentheses, it says blood path versus tissue contracting.
So this is where the chart comes in handy. So if you look at the difference in the chart between a blood path, same device, same as far as same category, external communicating, tissue bone versus blood, same length of time. If you look at the difference, the only difference between those two is hemocompatibility. And the reason why is because when you look at risk, everything in the tissue is the same risk as the blood. Now, the end results may be a little different if you're looking at like the chemical evaluation, which Sarah will talk about tomorrow. But the only risk from a biocompatibility standpoint that's different is that blood interaction. So we have to look at complement activation potentially. We have to look at thrombosis and hemolysis, because then you have direct contact with the blood, where when you're in the tissue, you don't. Circulating blood, you have contact with waste blood. So those endpoints need to be addressed with that type of contact compared to the tissue contact.
Okay. So our next one is, does the intended use also consider any information from a human factors usability study?
It may. So and once again, there's a lot of factors that could go into intended use. So when you look at human factors usability study, you look at how maybe if a user would not contact it because they're gloved or gowned or whatever it is, then you can help justify that based on that study that you don't have clinician use, and if it's not going to contact the patient, you don't have patient use. So you can use that. And there is a little bit of gray area from the FDA exactly how detailed they're going to get into that user because it's relatively new. Right now, we're really only seeing it for those devices that are intended to have skin contact for the clinician, like I said, masks and gowns and the headband on the protective shield and things like that. So those are the things we're seeing mostly come from that.
Okay, so let's see. Do people have success with the threshold for toxicological concern approach?
Yeah, in fact, we've been very, very, I hope you come back tomorrow because that's what Sarah pretty much is going to be talking about. But there are, depending on your regulatory arena again, there are certain ones that are more successful for the FDA than others. For example, genotoxicity, subchronic, subacute, chronic, carcinogenicity, all those endpoints are a little bit, a lot more easy to assess than sensitization or irritation. So there are certain endpoints that are easier with a threshold of toxicological concern, but yes, that is something that we use quite often.
Okay, great. So we are starting to run out of some time, so I'm going to give you about 2 more questions, Thor, and then we'll close it out. The next question is, since the guidance document, how frequently are you seeing the FDA come back and ask for carcinogenicity? Did I say that right?
Carcinogenicity, yeah.
Yes.
For permanent implant devices, we see it now almost, I don't want to say regularly, but we see it quite a bit. The question you usually come back, please perform a carcinogenicity evaluation using a threshold of or toxicological evaluation, or TE or TI. So they're not asking you to do the testing, but they're asking you to evaluate it, and we're starting to see that more. So my assumption is that if you have carcinogenicity marked on that new guidance chart, you're going to want to do chemistry, because that's the best way to get out of doing cancer, carcinogenicity.
Okay, great. You know, I apologize, but we are out of time. We will be giving these questions to Thor and Nelson to be answered via email. Thank you for asking such great questions to the audience. Thank you to Thor for an excellent lecture. This presentation will be available shortly in an on-demand format. As a registered user, you will receive an email with detailed information on how you can access the on-demand replay of this course. Please feel free to invite your colleagues and peers who may have not been able to listen to this event. This course is copyright 2016 by MD&DI. The presentation materials are owned by or copyright by Nelson Laboratories. The individual speaker is solely responsible for his content and opinions. I'm Tanya von Gremkow. Thank you for attending today's course, and be sure to join us tomorrow and Thursday. Have a great day.
Thank you for using the conferencing center.